Good morning. My name is Ross Chambers. I'm the Vice President of Antibody Discovery here at Integral Molecular. And what I want to share with you this morning are the lessons that we have learned over many years of generating antibodies against multipass membrane proteins. First, for those that don't know who we are, Integral Molecular is the industry leader in discovering antibodies against membrane proteins. We are a biotech company based in Philadelphia, solely focused on membrane proteins. We have almost 20 years experience of working with membrane proteins and have worked on many thousands of different membrane protein projects. We have developed deep expertise in antibodies, and we have developed an antibody discovery platform that's specialized for the unique challenges of membrane proteins. And this has proven highly successful at discovering antibodies against targets such as GPCRs, transporters, and ion channels. So Integral offers a number of different technology platforms to you around membrane proteins. The topic I'm going to talk about today is our MPS antibody discovery platform that goes all the way from targets all the way to lead panels of antibodies. We also offer lipoparticles that I'll describe in a moment. These are membrane protein preps that are useful for a number of applications. We have shotgun mutagenesis for epitope mapping membrane proteins. And we have the membrane proteome array, which enables specificity testing for membrane protein targets. All right, so first let's frame the problem. So as you may be aware that there are many thousands of multipass membrane proteins in the human proteome. Many of these are drug targets exploited by small molecules. However, from a antibody drug perspective, there have been very few antibody drugs successfully generated to these targets. In fact, there's only 3 of these targets have approved antibodies to them. The reason is, of course, these targets are very difficult to make antibodies against. They have a number of problems associated with them, and many of them have multiple of these problems, making it extremely challenging to make antibodies. A key one is the fact that these proteins, their structure is dependent on the lipid membrane. This extremely restricts the options for generating the antigen and manipulating it to ensure that the native epitopes are maintained. So conventional approaches applied to this class of membrane proteins is largely unsuccessful. Many of our customers who have come to us over the years applying conventional approaches either take many years of multiple approaches to generate antibodies, but generally with a very low success rate. The platform that we have developed here has a greater than 95% success rate and delivers it within under 1 year. Now, what we have learned over the many years of working on these kinds of projects is that there are many problems, as I outlined before, that you have to overcome to be successful here. Top of the list is a risk assessment that we start all programs with. And this is not always done with conventional programs, but I highly recommend it for this class of membrane proteins. So first up, you want to understand the underlying challenges that you have to solve in this program and come up with a mitigation plan to deal with them. The common problems that are identified that make these challenging is often expression. Many of these have very small extracellular regions, which makes them very poorly immunogenic. And many of them are highly conserved, which also makes them poorly immunogenic. Once you have identified the issues and have some kind of plan to deal with them, you then need to consider the antigen. And as I said, these proteins, their structure is dependent on them being embedded in the membrane to maintain the native epitope. So that restricts the kinds of formats that are available. So you have to think carefully about the antigen format. For a source of antibodies, we rely on animals. And we like animals because, one, they deliver a diverse range of antibodies. And also, one of the very powerful technologies with this field is DNA immunization. And that's only compatible with animals. When we get after that to, if you're successful at generating titers, Your MAb isolation has to be highly effective so that you get large numbers of antibodies and that you cover diverse epitopes. And then if you've done all of these steps correctly, the last step, lead selection, is pretty straightforward. You're able to find high affinity binders that are monospecific, functional if required, and they're easily humanized. So first, a few words about the underlying technologies in the MPS antibody discovery platform. So this technology platform is specialized for dealing with membrane proteins and has a proven success rate of greater than 95% at discovering lead panels of antibodies. The key parts of these technologies inside this are the fact that we optimize the antigen. We present it in the form of Lipoparticles and DNA, and we immunize into chickens to overcome the highly conserved nature of these targets. The B cells from those immunized chickens are then converted into immune phage display libraries, and we pan those on lipoparticles. To enable us to get large numbers of antibodies, we rely on automation so that we can screen through these libraries very deeply that have led us to discover, agonists and antagonists, rare functional antibodies. For engineering the antibodies, we're able to humanize these, which I'll show in detail later, affinity mature if required, and we do have a common light chain technology for generating bispecifics. So we have applied this MPS antibody discovery platform across a wide range of different membrane proteins across all classes, and have proven to be successful across all of these classes of membrane proteins. This table here is a list of our internal pipeline for the development of therapeutic antibodies, and I'll go through a couple of examples of these. All right, so first I want to go into some detail about overcoming some of these problems. So critical is developing an optimized antigen. The problems we're trying to solve here are low expression, and many of these targets suffer from low expression. They can be toxic to the cells that they're being expressed in. They can be unstable, or they could have regions of them that are immunodominant that are not relevant for the functional sites that you're pursuing. So the challenge here is to engineer or optimize this antigen while maintaining the native epitopes. And for that, we use this optimized expression plasmid. We use codon optimization. We will mutate the antigen itself, either delete regions or put point mutations in to solve some of these problems. And we will use translation and trafficking enhancers to get high levels of the protein to the surface. And really, this high levels of expression are key for getting that high immune response and for efficient antibody isolation. A few words about Lipoparticles that aren't familiar with these. This is one of the founding technologies of our company. So Lipoparticles are virus-like particles that are built from a viral core that contain membrane with high concentrations of the target membrane protein. These proteins are in a membrane, so they are presenting the epitopes in a native format. And being virus-like particles, they're highly immunogenic. When we immunize animals, we use a combination of DNA immunization and lipoparticle boosting. For those that are not familiar with DNA immunization, this is a process where you inject into the animal purified DNA plasmid. This transfixes the cells inside the animal, and the membrane protein is presented on the membrane in its native format for the immune system to recognize. Now, often this results in a mainly memory B cell response. So even though there's an immune response, you see no titer. So in order to efficiently see titer, you really need to boost these animals with a high concentration of the protein. And for that, we use lipoparticles because they are highly immunogenic and they contain high concentrations of the membrane proteins. And so it's this combination that's proven to be very effective at giving high titer responses reliably to these difficult membrane proteins. Now, as I mentioned, we often use chickens for our immunization. And the reason we have focused on chickens is that half of all drug targets are more than 90% identical across mammals. What this means is that many of the epitopes are unavailable because of immune tolerance. So we have moved to chickens as the immunization host because of the much greater evolutionary distance from mammals. Mammals as a group in general are very tightly related from an evolutionary perspective. So it doesn't make much difference whether you immunize a mouse, a rabbit, or a llama. You're generally going to have many of those same epitopes unavailable because they're highly conserved. So chickens enable us to get access to those highly conserved epitopes and also generate antibodies to epitopes that can react across different species. Some examples of the kinds of titers we get from immunizing with the system. So here's a subset of targets with various different classes of difficult membrane proteins. And all of these are highly conserved. They're more than 95% identical between human and mouse. However, the chicken identity is typically 10 to 15% lower relative to mice. And what this means is that these targets are robustly recognized by the immune system of chickens, and we reliably get high titer responses to these highly conserved proteins. So as I mentioned, chickens are also, a side benefit is that you can get pan-reactive antibodies. So you're able to access those conserved epitopes across species, which means that your therapeutic antibody doesn't require a surrogate antibody for preclinical studies. And what we have observed is that for many of these targets that are more than 90% identical between human and mammals, other mammals, is that we often see reactivity to rodent species and other mammals. So once you have an immune response, we then convert those antibodies into immune phage libraries, and we pan them on lipoparticles. And for panning, high concentrations of the target antigen is really key for efficiently enriching the hits out of the libraries. And lipoparticles have been very effective at doing that. Of course, we want to discover large numbers of antibodies, and so for that, we've relied on a lot of automation so that we can do many, many thousands of immunoassays to screen through and effectively find all the hits. And then for many of these targets where we need functional antibodies, we need to have them in an IgG format to perform functional studies. With that, we've developed an efficient system to enable us to subclone hundreds of these hits from the phage libraries and produce purified IgG that can feed into functional assays. Now, we have done many programs over the years where we've been tasked with discovering functional antibodies, and our platform has been very successful at doing that. Part of the reason may be for many of the reasons that I've mentioned before, but I also want to highlight one other feature that may be contributing to that success, and the fact is that chickens have longer CDRH3s. So in the graph in the middle, you can see in blue the lengths of CDRs of most therapeutic antibodies. These are mainly derived from mice, and that's why the average length is around 10 amino acids. Humans and chickens and llamas have longer CDRs, and what this means is that the paratope of the antibody protrudes. So typically antibodies past about, typically HCDR3s that are longer than 15 amino acids protrude from the surface of the antibody. And what this means is that those protruding loops, CDR3 loops, are able to access pockets on the protein. And many of those pockets are important for functional activity. And this has been well demonstrated in llamas, as shown in this figure here. Right, so you've generated a panel of antibodies. You then need to focus in to develop lead candidates. And for that, you need to produce the antibody, do some developability analysis, do some characterization such as epitope mapping, some functional assays, and some specificity testing. You may have to do affinity maturation, although typically we find that chickens deliver single-digit nanomolar to picomolar binders. And then you may have to humanize, and because we're using chickens, we need to humanize. Now, our experience with humanizing chickens is that they are very straightforward to humanize, and we and others have shown this. So typically, when you CDR graft the chicken CDRs onto human frameworks, you usually maintain almost all of the affinity of those antibodies. This stems from the fact that the chicken framework, even though chickens are evolutionarily distant, the actual antibody framework is highly conserved to humans. And the closest germline in humans is VH323 and VL325. And those two frameworks are well-behaved frameworks that are used in many existing clinical antibodies. Also making this task easier is the fact that chickens only have a single VH-VL framework. So our experience has been that this is very straightforward to do the humanization. And because it was so straightforward, this led us to take another leap and develop a new platform for humanization that humanizes them before isolation. So not humanizing them after you've found the hit, but actually humanizing the animals in the library itself. So this platform that we're revealing here for the first time at PEGS, this humanized chicken antibody platform, as I said, humanizes the antibodies prior to MAb isolation. So we immunize chickens as normal, but instead of pulling out the VH and VL, we only isolate the CDRs from the antibodies, all of the pool of CDRs from all of those antibodies. We then graft all of those CDRs as a pool onto a human framework in an scFv format in a phage library. That library then can be used as normal to isolate antibodies, but the key difference here is that the antibodies coming out of these libraries are already humanized and are typically high affinity. Now on affinity, one of the surprising features of this is because we're shuffling the CDRs, what we actually observed is that this results in affinity maturation. And this platform delivers reliably picomolar binders to these targets. To illustrate how well the humanization works, what we did is we've used this platform to discover antibodies to a number of targets. And here we've looked at the sequence comparison of these humanized antibodies to the IMGT database using domain gap align program. And what you can see here is typically these humanized antibodies coming out of this platform are between 91 to 95% matching to human, which is right on top of where fully human antibodies have identities to. Now, I mentioned that the shuffling of the CDRs that's intrinsic to this platform results in affinity maturation. To illustrate this, what we did here is after discovering a panel of these hits from this humanized chicken antibody platform, we went back and isolated the parental chicken antibody that led to these antibodies. For this, all these antibodies here have the same H3 sequence, but with different CDRs to the other parts of the antibody. And what you can see here is that we're able to isolate a large number of antibodies with improved affinity over that parental chicken antibody. To put that in some context, we are getting high affinity antibodies out of this platform, and here the lead candidates from two of our programs for the Claudin-6 and the Claudin18.2 therapeutic program, we have discovered antibodies that are either best in class or matching the affinity of a clinical benchmark. These are picomolar binders. Now, a key feature of antibodies, of course, is their specificity. And although many people assume antibodies are monospecific, it's been established that many antibodies, up to a quarter of antibodies, are actually able to bind other targets. To address that for membrane proteins, what we have done here at Integral is develop the membrane proteome array. This is a collection of essentially all human membrane proteins assembled in an array. These proteins are expressed in live unfixed cells, and we can interrogate that array using antibodies in flow cytometry to detect any off-target binding. And because flow cytometry is so sensitive, it's a very sensitive method for detecting any off-target reactivity. To make sure that the antibodies coming out of this humanized chicken antibody platform, we tested the antibodies on the membrane proteome array, and the antibodies coming out are indeed monospecific. They're very specific to the target, binding to their target, and not to any of the other 6,000 membrane proteins, or even closely related paralogs that only differ by a few amino acids. Okay, so that's the details of our platform. Just to summarize what I've said here today, Membrane proteins are very difficult to develop antibodies to. They require specialized technologies to be successful, to overcome the numerous issues that they present. And we here at Integral Molecular have developed a highly successful platform that are able to overcome these problems and successfully deliver antibodies. Working with us, we offer a number of reagents and services such as epitope mapping, the membrane proteome array for specificity testing, and Lipoparticles. We offer the antibody discovery platform as a partnership, and we have a number of therapeutic antibodies that are available for licensing. If you want to learn more about us and our technologies, we have a number of posters at PEGS, as well as Ben Durant's yesterday gave a talk on the membrane proteome array with more details about that. With that, I'd like to thank you for your attention, and I'll be happy to answer any questions.